Hello everyone and welcome to another session today for A&P. Couple of housekeeping things. Remember that when we see each other physically again on September 14th, we will have a quiz. After you take your quiz and we go over a quiz, we'll have a little bit of a break, and then we will go and take our lab practical. Lab practical covers all of Unit 1, and that would include all the histology and all those directional terms. You will also have due your worksheets, so your lab worksheet will also be due. Remember, I don't grade it for correctness, I grade it for completion, but I know that some of you all kind of had a few questions about some of the structures that we were looking at, specifically for like Part 4, where it has the directional terminology. A couple of hints. When we're talking about a cat, we want to use the terms ventral and dorsal. Another hint I want to give you is that there can be more than one correct answer for those terms. So I think one of them was about the navel and the hip bone. The navel in comparison to relation to the hip bone, or the hip bone in relation to the navel is, you could say lateral or you can say inferior. So there can be more than one answer for those questions, especially on part number 4, those lettered sections. There can be more than one correct answer. And also remember, when we're talking about a cat or an animal, we use the terms ventral and dorsal. We kind of reserve the terms anterior and posterior more for human species. So those are all of our little announcements. Your quiz, remember, is going to cover the lecture that we had on 8/31, I believe it was. I'm looking at my calendar here. The lecture that we had on August 29th, August 27th, and well, wait a minute, I think I went back too far. The lecture that we're having today, which is September 9th. The lecture that we had last Wednesday, which was September 2nd, and then the lecture that we had on August 31st. So that's what's your quiz is gonna cover, the August 31st lecture, the September 2nd, and today's lecture, the September 9th, will be what's on your quiz. All right, so before we finish up Chapter 5 here, questions, comments, or concerns about anything? And I have also received a lot of your emails about our exam and the dates that you can't do. And I have several students that are going to take it on Wednesday at 5 o'clock or in that time frame. So if you've emailed me about an examination and I have not emailed you back, please be sure to email me again, because it means that somehow I didn't get it, or somehow see me during my office hours, so that we can make sure that we get a time arranged for you. Okie dokie. So where we last stopped on this last Wednesday was that we were talking about how illnesses can manifest themselves as variations of skin color. And one of the first ones we talked about was jaundice. For babies, newborn babies that have a little touch of jaundice on there, we're not super, super concerned unless they're just really, really premature. They have a lot of other health conditions. But a lot of babies, I think, let's see, 2 out of my 4 children were born with just a little touch of jaundice, and a lot of times they'll put them under the bilirubin lights in the nursery, or they can even send you home on time with a billy blanket, which is just a little UV lights that you wrap around their little abdomen. And for my oldest daughter, the recommendation, the prescription was to just sit her in the sunlight for a little while every day, and that helps to break down that bile that causes the yellowing of the skin and of the whites of the eyes. Now, when you get to be an adult, when you're no longer a newborn, it's not because your liver and your body parts are just newly functioning. It's usually a condition where you have jaundice because there's some sort of liver failure. So your liver is not functioning because it's not super new, it's functioning because it's disease for some reason or another. We talked about Addison's disease, where it's what we call hyperpigmentation, so it causes the skin to have a darker coloration to it, and this is a disease of the pituitary gland. We'll talk about Addison's disease again towards the end of the semester in the endocrine system. And we talked about how JFK is probably one of the most famous people that you may recognize as an individual who had Addison's disease. And then vitiligo, which is hypopigmentation, it's on the other end of the spectrum from Addison's disease, is a loss of melanocytes. So those cells that are responsible for giving coloration to the skin, they are no longer there. So as a result, you see this hypopigmentation in various areas around the skin. The most notable person you may recognize that had vitiligo was Michael Jackson. So moving on to other parts of the integument. Remember, the integument is composed of two portions. We have the cutaneous membrane and the accessory structures. The cutaneous membrane consists of the epidermis, which is the layer of cells on top. There are 5 layers in thick skin and only 4 layers in thin skin. Thick skin we find where again? Where do we find thick skin? Palms of the hands and soles of the feet. Awesome, Rachel. So yes, that's where we find thick skin. And then thin skin, well, that's just everywhere else. I forgot to turn that thing on. All right. And we also talked about how the cutaneous membrane, which is one part of the integument, is separated into 2 portions. We have the epidermis and the dermis. The dermis, so we talked about the epidermis and how it's got those 5 layers, the stratum germinativa on up to the stratum cornea. The dermis now we're going to talk about has 2 layers to it. It's got what we have circled in red here, what we call the papillary layer. That is the most apical portion of this, or the top layer of the dermis. And then we have the bottom layer called the reticular layer. In that apical, or papillary layer, the top part of it, the type of tissue that you mainly find is a relar tissue. So if you remember from our histology discussion, a relar tissue is kind of that Jackson Pollock open framework, abstract art looking type of tissue. We have some very small capillaries that are in that region. So there we have some of those capillaries there, and the nutrients would, via diffusion and osmosis, go to the different layers of the epidermis, because remember, epithelial cells are avascular, which means they don't have a direct blood supply. You also have some lymphatics and some sensory neurons. So there we have that sensory neuron, the tactile corpuscle, right here at the top of the dermal layer. In your reticular layer down here below, you have more of a dense, irregular connective tissue. So it's not a relar connective tissue. Remember, with dense, irregular connective tissue has a bit more of an open framework. It was kind of the moderate on that spectrum of tissues itself. Lots of larger blood vessels would be found in this area. More lymphatic vessels, not shown in this picture, but lymphatic vessels would be running along in this area. Nerve fibers, not just the nerve, the sensory neuron itself, but the actual fibers would be found down here. And then you also may have some collagen and elastic fibers in the dermis itself. And the overstretching of those collagen fibers in the dermal layer is what leads to stretch marks. And we'll talk about those a bit more in a sec. So some things about the dermis, are issues we can have with, is what we call dermatitis, which is an inflammation of the top layer of the dermis, the papillary layer. And things that can cause dermatitis are an infection, radiation, mechanical irritation, so you're constantly having some friction with something that constantly rubbing against that area can cause this inflammation of that top layer. Chemicals, poison ivy, poison oak, those are all considered what we call contact dermatitis, because you come into contact with these different poisons. Some of the features of this inflammation or dermatitis is itchiness and pain, and there's usually some swelling that accompanies it as well. Along with your dermis, in your dermal layer, you're able to make what we call vitamin D3 or cocalciferol. Vitamin D3 is kind of a misnomer. Usually when we give something the name of a vitamin, we say that it's something that you have to get outside of your body. So the vitamin has to come from some sort of external source. Whereas with cocalciferol, vitamin D3, you don't have to get vitamin D3 outside of your body, because your body makes vitamin D3. So it's kind of a misnomer. Now, we do fortify a lot of our foods with vitamin D3. Milk and orange juice would be great examples of that. They can be fortified with vitamin D3. But in the presence of sunlight, your body will actually manufacture this. This is an excellent question for a short answer question, because this is actually giving a process of how does your body manufacture vitamin D3. So in the presence of sunlight, your epidermal cells are going to make this compound called cocalciferol. That cocalciferol is then going to make its way to your liver and your kidneys via the bloodstream. So the cocalciferol that's manufactured in the presence of sunlight, it's going to go diffuse into those capillaries or the papillary layer of your dermis, and then further down into the larger blood vessels of your dermal layer, and then on into your bloodstream and so forth. So it kind of makes its way to your livers and your kidneys, where vitamin D3 is converted into calciferol. The importance of vitamin D3 is so that it can aid in calcium and phosphorus absorption. We need that calcium and phosphorus absorption in order for your bones to maintain their hardiness. So this concept of vitamin D3, we will come back again when we go into our next chapter, the skeletal system. But in order for the calcium and the phosphorus to be properly absorbed and become the calcium phosphate or hydroxyapatite that is responsible for the hardiness of the bone, it gives them their, makes them nice and hard, we require vitamin D3 for that. If you have insufficient vitamin D3 that's either made or ingested by the body, it will cause those bones to sort of bow out and be much weaker than they normally should be. And that bowing out of the bones is what we call rickets. So understanding this process is a great short answer question, and I could ask something along the lines of, what is the importance of vitamin D3? Explain how this process takes place with sunlight in your epidermal cells. So as I said before, insufficient vitamin D3 can cause rickets, or as my grandmother would call it, the rickets. And she always told us, and my grandmother is from Mobile, Alabama, which is as far south as you can get in the state of Alabama without being in the Gulf of Mexico. So she's from the deep, deep south. And she would always tell us, and keep in mind, in the summer it's like really, really hot. And we're from up north. We're from northern Illinois, where we only really get like an 80-degree day was like a scorcher for us where we were from. So when we went to go visit my grandmother in Alabama, she would make us go outside and play. And she would tell us, you better go outside and play outside or you're going to get the rickets. Well, she was partially correct in that. We really don't see children, at least not in the United States and more industrialized countries, that have the rickets anymore because so many of our foods are fortified with vitamin D3. And now that I look back and I understand this, she was right. She had a point. Where she grew up in Marion, Alabama, they got their milk from Bessie the cow. And Bessie the cow's milk was not fortified with vitamin D3. They didn't have that fortification in it. So just drinking milk alone was not going to be enough to give you the vitamin D3 that you needed because it wasn't in the milk. So if you didn't go outside and play, if you were kind of sickly and you just didn't go out to play very much, then you saw these children that weren't getting adequate sunlight have this bowing of the legs, which was called the rickets. So she always said, if you don't go outside and play, you're going to get the rickets. And we had no idea really what the rickets was, but it sounded like something you didn't want to get, so we played outside. Dermal skin damage, a little bit we talked about already. Stretch marks typically happens from excessive weight gain and weight loss, and that's just the pulling of those collagen fibers in the dermal layer, the reticular portion of the dermal layer that get pulled and pulled and pulled, and then they just don't snap back like they used to. Weight gain does the same thing, and you lose that weight. Pregnancy is basically massive weight gain, massive weight loss in a relatively short amount of time. Whether or not you get stretch marks from weight gain or weight loss or pregnancy is completely genetic. Some people just don't get stretch marks. It's just the way that cookie crumbles. Some people do get them, so it's all completely genetic. And what I have found is that no cream, slab, anything that you put on it, if you're just genetically predetermined to get stretch marks, is going to make them go away. They're going to kind of always be your personal reminders of where your body's been. Sagging and wrinkles can be caused by dehydration, so make sure you stay well hydrated at all times. Age, obviously, the collagen underneath your skin starts to lose some of its elasticity, so as you begin to age, you may start to experience some wrinkles. I personally reject that notion, so that's not going to happen for me. Hormonal changes in your body, so accompanying age, you definitely have some hormonal changes that can also affect the elasticity in the dermis underneath your skin and your collagen fibers and your elastic fibers and the abundance of those fibers. And then ultraviolet light, so UV exposure. Another great reason, other than the prevention of malignant melanoma, another great reason to wear your sunblock when you're going outside. A lot of makeup and foundation for women or people that wear makeup has UV or SPF protection in it to prevent wrinkles that come too early, premature wrinkles. So definitely wear your sunblock. And if you wear a foundation or even just moisturizers, a lot of moisturizers already have SPF in them. And no matter how darkly complected you are, it's never a bad idea to put on some sort of light UV protection just to preserve your skin and to save it from damage and also from melanomas. Also within your dermis, you have what we call lines of cleavage, and these are parallel lines that will remain shut when the wound heals. If you make a cut that is parallel to your line of cleavage anywhere on here, then when it heals, it's going to be nice and secure in there. Why that's important to you as a practitioner is that you want to kind of have an idea of how the lines of cleavage are running so that when you perform surgery on a patient, that when the wound heals, it will have minimal scar tissue, and it'll be less likely to leave scarring or to pull apart. If you make a cut at a right angle across a line of cleavage, it has a tendency to produce more scar tissue, and it may have a tendency to pull apart. So now below the dermis, we have the hypodermic layer. It's underneath the integument. There's more elastic tissue here and areolar tissue and fat, so more loose connective tissues you'd find in this area underneath the dermal layer, so right down here. You also have what we call the cutaneous plexus, which is just a network of blood vessels in that area. You don't have any vital organs in your hypodermic layer, but this is also the site of what we call subcutaneous injections that require hypodermic needles. So one of the few accessory structures that we're going to talk about, hair and nails. Remember, in your textbook, it has this whole litany of items and things you should know about the hair and the nails, and I'm not going into that detail. So only what we discuss here on our Collaborate sessions is what I want you to know about the hair and the nails and the glands for those accessory structures. So those are the three accessory structures that we're going to hit on. The job of the hair is for protection and insulation. Another job of the hair is that it guards openings against particles and insects. Even in your ear, you have little hairs that stick out in there. The job of those hairs is that for any little buggies or any particles that are trying to get into your ears, they get stuck in those hairs as well with your earwax. So it helps to prevent things from getting inside your body. If you look in your nostril, you've got hairs there, so that opening that it's protecting is keeping insects and particles from getting into your nose. Your hairs are very sensitive to very light touch, and we talked about this in our first part of these notes, that if a mosquito lands on you and it lands on one of your hairs, sometimes you're able to swat that thing away before it gets a chance to stick out its little proboscis and take some blood from you, because you can feel that mosquito landing on you, and that you can perceive that very light touch. And you've probably noticed if even just one hair in your head moves, you can feel that. So some aspects of the hair and the hair shaft. We have sebaceous glands. The job of sebaceous gland is to lubricate the hair, and then it also contains sebum, sebaceous gland. So sebum is the liquid that helps to control bacteria, so it has an antimicrobial aspect to it. We also have the erector pili muscle, which is a smooth muscle. All of the smooth muscles in your body, they're under your involuntary control, which means that you cannot make them contract just by thinking about them. It's a different part of your brain that controls the contraction and relaxation of any of these smooth muscles. Other places you find smooth muscles in your organs, such as your esophagus and your stomach, those are places of smooth muscle, your uterus, etc. So for this particular smooth muscle, it's what it causes your hairs to stand up on end, or what produces goosebumps. So when this muscle contracts, it will produce goosebumps, and it's called the erector pili. And then we have the hair shaft itself. The hair shaft is not directly attached to the integument. However, at the hair root, it is attached to the integument. So this is well below the surface of the hair. We're down here past the epidermis and into the dermal layer where we find the hair root. The types of glands that we find in the skin, well, one we've already talked about, that sebaceous gland, or your oil glands. Not only do you have a sebaceous gland that's attached to your hair, but you also have oil glands all over your body, and some of us just make more oil than others. The type of gland this is, the type of secretion we have for these sebaceous glands, and this is also a very important concept to remember. Remember back in Chapter 4, we talked about the various different modes of secretion between holocrine, merocrine, and apocrine secretion, where here are some examples of glands that produce this sorts of secretions, or how we get this stuff out of the gland itself. So for holocrine secretion, remember the entire cell ruptures in order to get the secretin, or the thing, out of the cell. Oil glands are an excellent example of that. So they will secrete sebum by causing the whole cell to rupture. For your sweat glands, you have two different types, apocrine and merocrine secretion glands. The apocrine glands, those are what we call our stinky glands. So for the apocrine secretion, that would require the top of the cell, the apocrine portion of the apical portion of the cell, to come off. And we talked about how mammary glands sort of work that way. So the top of it comes off, and that's how the secretin gets out. So we find apocrine glands in your armpits, around your nipples, and your groin, kind of your stinky places that have a tendency to get stinky. Interestingly enough, it's your stinky places that people are attracted to or not attracted to. So kind of your pheromones are in these different, come from these different glands. Merocrine glands are more widely distributed. These are the ones that are responsible for that sensible perspiration, sweating, cutaneous evaporation, whereas we talked about how we had insensible perspiration, where your body just loses water off to the environment, but you're not really sweating. It doesn't make sense that you're losing it, but just because of the laws of diffusion, we have water leave off. So the merocrine glands are responsible for what we call sensible perspiration, and that cools the skin, or what your book, and what you've probably heard of it being called as well as cutaneous evaporation. So definitely water and some salts. You ever lick something sweaty, or yourself when you're sweaty, it tastes salty. So those are electrolytes, so sodium, chloride, potassium, things that you lose off. And then another job of these glands is that they help to flush microorganisms off of the skin and any harmful chemicals. So it helps to kind of flush the skin of anything that could possibly go systemic and get inside. It kind of has an antimicrobial role or function to it. So when we need to repair the integument, if there's been an injury to it, first thing that happens is that bleeding is going to occur. When that bleeding occurs, that's going to trigger these mast cells, and mast cells are denoted by this little guy here. It's going to trigger these mast cells to undergo inflammation. And as a result of that inflammation, they're going to release chemicals that will cause other cells to come to the area in order to try to heal it. So you have a scab that forms on top eventually, and then you have germinative cells that were called to this area by the mast cells through that inflammation or inflammatory response, that are kind of rebuilding the dermal layer and rebuilding the germinative layers of the epidermal layers. And they continue to do this and surround around the wound. So these migrating epithelial cells will come all the way down to the base of the wound, and they'll just grow and push up. All the while, you have more mast cells that are releasing prostaglandins and other chemicals to cause cells to come to the area to clean up the infection and to regrow that dermis and that epidermal layer. Macrophages are immune cells that will come to the area and they will clean it up, so they'll get rid of any of the excess debris or microbes or anything that was in the area that shouldn't have been there. And what you're going to be finally left with is what we call granulating tissue or scar tissue. So fibroblasts, endothelial cells, they kind of move in and they will produce this scar tissue or granulating tissue as a way of repairing the integument. And notice that even once the scab is gone, sometimes it leaves a little bit of a mark there because the integument may not repair itself in the exact same way that it was prior to the injury. This is also a good example of a short answer question because it's a stepwise process of what happens. And I could ask something simply as, explain how the integument repairs itself. Be sure to discuss the role of mast cells, macrophages, fibroblasts, and what is granulating tissue. So, and you should be able to tell me stepwise how that happens. For your short answer questions, I'm not really picky that you answer in essay form. I just want you to give me all of the information I need to show me that you understand this process. So if drawing it out and then labeling what you drew and explaining in a short paragraph what you drew, if that helps you to better visualize and explain the process, then that's okay with me. If writing it in bullets or charts or however you want to express yourself, you have creative expression to answer your short answer questions. The only thing that I answer is that you answer the question fully and that you demonstrate that you understand the concept. And this is for your lecture exams. Remember, quizzes, 15 multiple choice questions. And then finally, the types of injuries that we can have for the integument. These would be good questions for short answer questions as well, to describe the different types of injuries to the integument and perhaps give an example of one. So an abrasion is just scraping away the superficial layers of skin or mucus. So if you just scrape your knee when you're riding your bike, that would be an excellent example of an abrasion. A laceration is a jagged wound or cut. So that is something where instead of just scraping your knee and just taking off that top layer of skin, that you actually cut deeper into the integument down to the dermal layer perhaps, so you've actually been cut. One year my dad, we were in Texas, we drove to Texas, and he was trying to squeeze between two cars and he cut his knee on the license plate, so he had a laceration, which by the way he sewed up himself. It was a holiday weekend and he didn't want to go to the emergency room, so we went, I forgot how we got this, but somehow he ended up getting a suture kit and sewed himself up. It was quite fascinating. A puncture is an injury that's caused by a sharp, narrow object that's penetrating the skin. I remember my little daughter, Kira, when she was about 5 years old, she was upset because she had to go get a shot, and she said, they're going to puncture my skin. I don't know where she got that terminology from, but she was absolutely correct because it's a sharp, narrow object, and that's, you know, when you get an injection or vaccination, that would be a puncture. Straight in and usually coming straight out. An incision is a cut produced by a surgical instrument. Notice that a laceration, those are usually accidents. An incision, you did that on purpose because we're using a surgical instrument to create an opening. So incisions, we do those on purpose. Hopefully it's a healthcare professional that's making this incision. And a laceration, well, those are more, it's a cut, but it's more jagged and the edges aren't clean, and those are typically accidents. A contusion is just a fancy way of saying a bruise. So an injury that does not actually break the integument itself, but it causes swelling and discoloration and pain, and what we call a bruise. All right, questions, comments, or concerns on any of that so far? All right, so I think what we're going to do now is just take a little bit of time to start the next chapter. So I think we're going to go ahead and start a little bit of the bone structure and histology. So we're going to go ahead and I'm going to open up the next set of notes for us. For our next unit in lab, we're going to start to go over the muscles and the bones. So this is a good segue into what we will be doing on Monday in lab, or not this Monday in lab, but the next Monday in lab. So we're going to start talking about the skeletal system. All right. So bone structure, histology, and articulations on here. This is in Chapter 6, but remember your page numbers are going to be a little bit off if you have the newer version of the textbook. But what you want to look for is that it's going to start with the introduction of the skeletal system. So that's what, and I'm not sure exactly what the page numbers are in the new book. I don't have the new book in front of me right now. So your skeletal system, like your integument, consists of two basic portions. You have your bones of the skeletal system. You have about 208 bones, or 206 bones, give or take. Some people have one more, some people have one fewer. And then you also have what we could kind of consider accessory structures, but you have your different types of cartilages, ligaments, and connective tissues. All the types of cartilages that we find in the skeletal system and connective tissues, we've already learned about them in Chapter 4. So that's what made Chapter 4 so very important, is because it laid the framework for the rest of the structures that we'll talk about, because the rest of your organ systems are going to have, in some degree or another, epithelial cells and different types of connective tissues. And once you understand the jobs of those connective tissues, and you understand the jobs of those different types of cartilages and whatnot, you're better able to have an idea about how the different organ systems work. So jobs of the skeletal system, obviously for the support, so the bones itself are going to provide the internal framework of the body or support those muscles. The muscles allow the skeleton to move, but it's the skeleton that gives that internal framework. It also stores minerals in the bone itself, and specifically calcium. There's the storage of fat or lipids in the yellow marrow. On the inside, or the medullary cavity of your long bones, you have red marrow, and it's responsible for blood cell production, erythrocytes, and lymphocytes. Your bone also helps to protect various organs. Your heart lies right behind your sternum and your chest, so it helps to protect that very important organ, as well as your rib cages help to protect your lungs. And it also provides leverage for your muscles to contract again to provide that force of motion. So all of these are examples of the jobs of the skeletal system. The way that we classify bones is by three very basic categories. We can classify them by shape. We have bones that are considered long bones, so the bones of your arms and your legs would be long bones. We can characterize them by internal tissue organization for them, whether they have what type of spongy bone, or is it compact bone that's on the inside of it, or we can classify bones by their specific markings. So some of their surface features that we find on them, we can consider that a way to classify them. In our unit for Lab 2, our lab practical, we will talk about foramen, holes, and we'll talk about fossas, where they are shallow depressions. And these are places where bones may articulate with other bones, or some of these foramen may be where blood vessels and nerve bundles can go through. And by looking at those very unique markings, we're able to determine one bone from another. We're also able to tell where the bone articulates with another bone. So for your lab practicals, it would be very important to understand, and you're going to have to know it for your lab practicals, what are all these different little features on the bone and what do they mean? Those bone markings can also tell us the approximate age and gender of skeletal remains. So the primary classification that we're going to use in lecture for classifying your bones would be based on shape. So we have a long bone here, as I said before, found in your limbs, your arms and your legs. So your femur would also be considered a long bone, as well as your humerus. We have flat bones that make up your skull. So you have two parietal bones, a frontal bone, and an occipital bone, and you have temporal bones in the side of the skull. You have sutural bones, which are little teeny tiny, looks like bone chips that are just embedded along the suture lines of your skull. Irregular shaped bones would be like those of your invertebrae. All of your invertebrae are sort of shaped alike one another, but they're not shaped like any other bones in in your body. So we give them that classification of irregular. Short bones, such as in your carpals bones and your metacarpals and your feet and your carpal bones in your hands. Those are little short, squatty, stubby bones that we find there. And then sesamoid bones, the best example of a sesamoid bone would be your patella over your kneecap. And it kind of looks like an oversized sesame seed. Remember before I said people have about 208 bones in their bodies. Some people have a couple more, some people have a few less. It's usually sesamoid-shaped bones that they may have an additional bone in a joint somewhere. So for long bones, as the name suggests, they are long and thin, and as we've already established, found in the arms, legs, hands, feet, fingers, toes, all of those places. Even though the fingers and the toes are shorter long bones, they still have the same sort of configuration. Flat bones are thin and have parallel surfaces like we'd find for the bones that make up the skull. Your sternum is considered a flat bone as well. There's a parallel surface of compact bone in the front and a parallel surface of compact bone in the back. Your scapula and your ribs are, like your shoulder blades back there, those are all considered to be flat bones as well. Sutural bones, remember we only find those between the flat bones of the skull, so you have they lie along the suture lines of the skull, and they're just really small, kind of funky shaped little bones along those suture lines. Irregular bones, your vertebrae is an example of that, more complex shapes to them, and in your third lab practical, you all will tell me about those different facets, those articulating facets that are on those vertebrae. Your pelvic bone is also an irregular shaped type of bone. Short bones, small and thick, wrist and ankle bones, sesamoid bones, small and flat. Most prominent is your patella, but they can also develop inside near tendons, of the joints of hands and feet, as we said before. If you have, some folks have 209 bones, and sometimes they have an additional bone, and a sesamoid-shaped bone in your hands or feet or any of those joints near those highly synovial joints. So those bone markings that we talked about, depressions or grooves. A depression would be like a fossa is a name for a depression or a groove that you'd find in your bone. And this discussion that we're having now, we'll come back to it again when it's time for our, when we talk in lab class. We'll talk about them again for Unit 2. But those depressions or those grooves are certain types of bone markings. A lot of those depression or grooves are places where blood vessels will go through, or you have bundles of nerves that can go through there. You have projections that are also found of your bone. Things that we consider like a condyle or an epicondyle, those are all projections that will protrude from it. Foramens are sort of tunnels where blood and nerves enter the bone. So these are different markings. Depressions, projections, and tunnels, these are all markings that we would find on the bone itself. As we go through this chapter, what I want you to notice is that the first thing, and actually all of your chapters in A&P are kind of set up the same way as using this Martini book, that first we're going to take a tour of the organ system. We're going to look at what types of structures are there, what types of cells are there, what types of, in this case, ligaments and tendons or whatever else it may be. What are some things that we would see on this tour? So if you ever liked the Magic School Bus growing up, or if your children still watch reruns of the Magic School Bus, then you can appreciate this. I'll be like, what's her name, Miss Frizzle, Miss Fritz, then I'm going to be your tour guide. So we're going to look at all of these aspects and portions of the skeletal system, of the organ system, and then once we have a firm understanding of what these different parts are that make up this organ system, then we're going to turn it on. Then we'll talk about exactly how these structures work together in order to achieve its function. So next up on our tour is that we're going to look at a long bone and talk about how a long bone has very distinctive portions to it. The first portion of the long bone is that it has that long shaft. And we call that long shaft the diaphysis. So that like diameter, means to go through. So the diaphysis goes through and is the longest part of the bone itself. So I'm going to attempt to draw this. Bear with me. So this portion here, that is our diaphysis. That's the shaft of it. The outside of this shaft is composed of very heavy, compact, or what your book also calls a dense bone, but we're just going to use the term compact bone. On the inside of this shaft, we call that the medullary cavity, and it can be filled with different types of things, and we'll get to that later on. Your epiphysis, on the other hand, are the knobby parts of the bone. So the wide parts at each end are called your epiphysis. And this is the point where the bone is going to articulate with other bones. Within this epiphysis, on the inside, you have spongy bone. So the spongy bone lies on the inside of the epiphysis, or at the ends of it. On the outside of it, just like the shaft, it's going to be covered with compact bone. For the metaphysis, it's where the epiphysis and the diaphysis meet. So that's just going to be where they meet. Your metaphysis is also where your growth plates are found. So that cartilage that's constantly being converted into bone as you're growing, that's at your metaphysis, between your epiphysis and your diaphysis. And here's a much nicer picture of what I attempted to draw. So we have the compact bone that's surrounding that empty or hollow cavity, that central cavity, that medullary cavity in there. And then you have your epiphysis on the end of it, and they're filled with compact bone. So this is an actual bone that we're looking at a longitudinal cross-section of. Now for flat bones, like the parietal bones of your skull, remember you have two of them, you got a left and a right one, they're going to resemble more of a sandwich-like configuration where there's compact bone on the top and compact bone on the bottom. That would be like the bread. And then the inside of it is going to be what we call a diploe. Diploe is a very high-resistant form of spongy bone. So this diploe will be able to resist stress in multiple different directions. So that's one of the reasons why it's pretty difficult to crack your cranium or to fracture your parietal bones or bones of your skull, which is a good thing because you got a very important organ that lives inside of your skull. It's very difficult to fracture them because of this diploe. This diploe is able to withstand stress in many different directions and dissipate that stress over the layer or the surface of the compact bone to keep it from just breaking in one spot or fracturing. Beautiful picture of this. We have a compact bone on top, compact bone on bottom, and then this diploe is basically like spongy bone on steroids. So notice that the struts here are much thicker, much wider, so that when force hits the compact bone of this flat bone, that force is going to be dissipated along the surface of the compact bone as a result of this diploe. So the type of tissue that bone is made of is we call it osseous tissue. If you'll remember from Chapter 4, we talked about how bone is a type of, well, is it an epithelial tissue or is it fall under the umbrella of connective tissue? Bone is connective tissue. So things that we know about connective tissue is that it has specialized cells, it has a fluid portion of it, that ground substance, and it's got protein fibers. So for this osseous or bone tissue, there are, because it is a dense supportive connective tissue, it has specialized cells that we're going to talk about. They're 4 different classes of cells: osteoprogenitor cells, osteocytes, osteoblasts, and osteoclasts. So those are the specialized cells that make up the bone. It also has a solid matrix of calcium salts. So we're going to talk about the calcium salts that are in there. Not a whole lot of ground substances, mostly protein fibers that make it up, and you have these calcium salt deposits. And we also have protein fibers, and the protein fibers that we find in this osseous tissue are collagen fibers. So far in the skeletal system, all we've done, we haven't talked about exactly how any of it works. We're just taking a tour of what you would find in the skeletal system. So we've talked about how you'd find long bones, flat bones, irregular shaped bones, sesamoid bones, etc. We talked about how all of these bones are going to have some sort of marking or configuration to them. They'll have depressions or fossas or epicondyles or condyles or foramen, tunnels or grooves. They'll have all these different portions to them. We haven't talked about what those things do, but we just talked about this is what they are. And now we're starting to get down into a cellular level and say that yes, bone is a type of connective tissue. And just like all the other connective tissue, it's got specialized cells, ground substance, and protein fibers. Ground substance and protein fibers make up the matrix. As we move further, we're going to talk about the specific kinds of cells that are specialized and what they do. So, characteristics of this bone tissue that we're talking about. Very, very, very dense matrix. So what that means is that there's very little fluid that you would find as part of this connective tissue, which makes sense that we shouldn't have a whole lot of fluid here because it's structured for support. Osteocytes are the actual bone cells, and the osteocytes, which are in the lacuna, are going to organize themselves around the Haversian canal, or the blood vessel that comes into the bone. The cytoplasmic extensions that extend out from the osteocytes are called canoniculi. They form pathways for the blood vessels so that the different osteocytes can communicate with one another. And all of this, when I show you the picture of it, we've talked about and seen in lab. And then you have the periosteum. Peri means around. So the peri, and then osteo means bone. So the periosteum is going to cover the outer surfaces of the bone. And that periosteum has two portions to it. The outside, more fibrous portion, and then the inside cellular levels. So once again, we're just taking the tour. Other aspects of that bone, that osseous tissue, so we've already talked about some of those specialized cells. The osteocytes would be those specialized cells, and then we just talked about the parts of those osteocytes, how they have the canoniculi that mix up their cytoplasmic extensions and how they're able to communicate with one another. And we talked about the surrounding, the fibrous connective tissue that surrounds the bone itself, or that periosteum. Now we're going to talk about the matrix, so the ground substances and the protein fibers. Within that matrix, most of it, in fact 2/3, that's a number I want you guys to remember, is called calcium phosphate. That calcium phosphate reacts with calcium hydroxide to make hydroxyapatite. This hydroxyapatite is what incorporates those calcium salts and those other ions, and it helps to give the bone its strength, to make it nice and strong and hard. A very cool segue here, or connection, is that in Chapter 5 we talked about how vitamin D3 helped with calcium and phosphate absorption. This is the calcium phosphate that we're talking about that is absorbed in the skeletal system as a result of vitamin D3. The proteins that make up this matrix are going to be collagen fibers. 2/3 of the bone matrix is calcium phosphate. 1/3 of the bone matrix is just these collagen fibers. So the job of the collagen is to kind of give the bone a little bit of give, if you will, so it doesn't make it so super, super brittle. Without the collagen fibers, the bone would be very, very brittle and would break or fracture or shatter very easily. It is the collagen that helps it to have a little bit more bounce or give to the bone so that it doesn't shatter upon impact all the time. The calcium phosphate, its job is to make sure that the bone is nice and hard. If the bone was just consisting of only collagen fibers, it would be very floppy and very flexible, but it wouldn't be able to withstand a lot of body weight. So both portions of the matrix are equally important. 1/3 collagen, 2/3 calcium phosphate. other cells that make up the bone. So we have those specialized cells, perimeter connective tissue, 3 parts to it. You've got ground substances, protein fibers, both of which we talked about, and then you have those specialized cells. One of those specialized cells we talked about were osteocytes. These specialized cells only make up about 2% of the bone mass itself, but we have osteocytes, which are the actual bone cells. Osteoblasts, these are the bone building cells. Those osteoblasts can become osteocytes. Osteoprogenitor cells are your stem cells that can become osteoblasts, and osteoclasts, these are what I like to call the bone crashing cells. These are cells that will liberate calcium and put them into the bloodstream if your calcium levels get too low. So osteoclasts will break down osteocytes. Osteoblasts will build, B for build, blast, will build osteocytes. And osteocytes, well, they're just the bone cells that we find with canaliculi around the central canal of contact bone. So osteocytes are your mature bone cells. They live in the lacuna, and we talked about a lot of this in lab, so I'm not going to spend a whole lot of time on it. They are between what we call lamellae layers. They have canaliculi that will extend to communicate with the other bone cells. And a very important feature of osteocytes is they don't divide. When it's time to make a new osteocyte, an osteoblast will be triggered through hormones and various other entities to become an osteocyte. But osteocytes don't continually divide. Jobs of osteocytes are to maintain the protein and mineral content of the matrix, and also to help repair damaged bone when osteoblasts become osteocytes. Osteoblasts are our bone-building cells, B for builders, B for blasts. They secrete an organic matrix that we call osteoid, and the osteoid that's produced by the osteoblast kind of encirculates that osteoblast until calcium and phosphate comes to fill in those areas or deposit it into that osteoid. As an osteoid is primarily made out of collagen fibers. So the hydroxyapatite and those calcium phosphate minerals will calcify around the osteoblast, and as that happens, as it becomes surrounded by bone, the osteoblast becomes an osteocyte. So osteoblasts are your bone-building cells, because if you will remember, osteoclasts don't divide, or I'm sorry, osteocytes don't divide. So when you need new osteocytes, you have to build them from the osteoblast. Osteoprogenitor cells are a special kind of mesenchymal stem cells, which we've already discussed how mesenchymal stem cells are one of the 9 cell types that you can find in connective tissue. They divide and become osteoblasts. Where we find osteoprogenitor cells are in the endostinum, or the inner layer, the inside portion of the bone, the endostinum itself, and the inner cellular layer of the periosteum. The job of the osteoprogenitor cells is to repair a fracture, because the osteoprogenitor cells produce osteoblasts. Osteoblasts secrete an osteoid matrix that eventually gets filled with these calcium and phosphate mineral deposits, and as that area calcifies, those osteoblasts then graduate to become mature osteocytes. So that's how the osteoprogenitor cells becoming osteoblasts, who become osteocytes. That is that whole process of assisting in fracture repair. And then our last type of bone cell are osteoclasts. Osteoclasts are going to be the bone crashing cells. C for crashing, C for class. They're going to break down, they secrete acids and protein digesting enzymes to dissolve the matrix and release the calcium and the phosphate that's been stored on the outside of these osteocytes, these mature bone cells. And we call this process osteolysis. The stem cell that osteoclasts come from is completely different from that mesenchymal stem cell that osteoblasts arise from, or osteoprogenitor cells. It comes from a different stem line than, let's just go back, this guy. This is the stem cell that produces the osteoclast. The stem cells that osteoclasts arise from are more like macrophages, because they have this digestive quality to them. So there's a fine balance between bone building and bone breaking. So the bone building by osteoblasts and bone recycling by osteoclasts, there's a nice balance between them. If you have more breakdown than you have building, if you have more crashing than building, then obviously the bone's going to become weak. And things that can cause that are poor diets, malnutrition, and age. It's something that just sort of kind of happens to a lot of people as they get older. However, exercise, specifically weight-bearing exercises, is going to stimulate more osteoblast activity. In a nutshell, what happens is that when you engage in weight-bearing exercises, it's telling your body that, hey, I need this bone to have more width to it, to be able to withstand more pressure, to withstand more weight. And so it causes the bones to become thicker, what we call oppositional growth. It becomes a lot thicker as a result of these weight-bearing exercises. So it's kind of your body's way of saying, well, if you're going to be doing these sorts of exercises, I better make sure that I prepare for it. And over time, you'll see more osteoblast activity happening to build the bone outward, not longer, but outward, so that it can withstand these different types of exercises and routines. All right, so that's where we're going to end for today. So everything that we've talked about for today's lecture, for last Wednesday's lecture, and last Monday's lecture on August 31st, that's all game for your quiz that happens on Monday. All right, so I think that's a good place to end. Questions, comments, or concerns about anything? All right, well, enjoy this wonderful Wednesday. It's a little rainy outside, but we needed a little bit of rain. And I hope you guys had a really good 3-day weekend. I hope you did something fun as like the last blast of summer. And I will see you guys on Monday well prepared. And remember, for your worksheet that's due on Monday as well, for Part E, a couple of hints to that. A, there can be more than one answer. And for your Part 4E, 4A through I think it's J, that when we're talking about animals, we want to use the terms ventral and dorsal for the cat question, so that might help. And if you still have issues, come see me in my office hours or shoot me an email, and we'll see if we can't get it worked out through that. I am in a new office right now, so I have a new office phone number, and the extension is 7713 if you want to call me, if that's better for you, to call me during my office hours. New extension 7713 if you want to talk about your lab worksheet. For the other parts of your lab worksheet, like labeling those organs and labeling the histology, if you use the images that are on Blackboard for histology and you use your textbook as a guide, that's more than sufficient for a key. I think it's just that very first part of the lab worksheet that people kind of had a little bit trouble with answering those questions properly. All right, so if you guys don't have any other questions, comments, or concerns, enjoy your day, and I'll be talking to you soon. Bye.